your host, Jim Gall. And thank you, Cari, for turning that on again. This is being recorded, just so you know, so that if you have friends who can't make it tonight, the video link will be up under. If you just Google Grit City Think and Drink, you'll find the link to all this talk and the previous talks that have gone on. I'm your host. I'm a professor at the University of Washington, Tacoma, in the School of Interdisciplinary Arts and Sciences. And I have been organizing this thing for a few years now, and I'm glad you all joined us, even on this amazingly nice weather day. But as we know in the Pacific Northwest, anything over 70 degrees is too hot. So I'm glad that you got out of that heat to come in and listen to our awesome speaker tonight. We do this every month on the second Tuesday of the month. So Cari is going to give us an awesome talk, and I'll introduce her just in a second. But our next talk is coming up in August, on August 11th, and that's Jim Thatcher at UW-Tacoma, who's going to talk about gerrymandering. So I hope you all will tune in for that as well. And we have talks planned all the way through the end of the year and beyond. So please come and check out all our different things that are going to be going on. The way this works, for those of you who have not been here before, or been here virtually before, maybe that's the way it is that Cari's I'll introduce Cari. She'll give an awesome talk, and then actually normally we would give away some stuff right then, but Cari has some questions embedded into her talk tonight. So we're going to ask you to chat your answers, and the person that gets closest on 2 of those questions actually wins the amazing Think and Drink socks. These are limited edition. Once they're gone, they're gone. We've gone through Think and Drink shirts, Think and Drink hats, and we're now onto socks. So eventually, next year, if you tune in, we may be onto underwear. So I hope you all will all come join us for that. And we will also have a question and answer at the end. So if you're thinking up questions, you can chat those as well. And then I will kind of go through those and feed them to Cari, and she can answer your questions. So we'll play Stump the Speaker towards the end of this thing. So thank you all for coming. And now, without further ado, I want to introduce our speaker. And I will stop sharing my screen so that Cari can share hers. Cari LeRoy has been a freshwater ecologist at the Evergreen State College since 2006. She completed her PhD in biological sciences in 2005 at Northern Arizona University in Flagstaff, and a master's in liberal studies, environmental education in 2001, and her undergraduate degrees in environmental science and international studies at Oregon State University. She co-directed the Sustainability in Prisons project from 2011 to 2017. And her research focuses on how riparian forests interact with streams and provide energy through leaf litter fall, or the stuff that basically you have to rake up in your yard every fall. Her research has shown that both the species diversity and genetic diversity of these litter inputs can affect in-stream leaf litter decomposition rates, aquatic fungi, and aquatic macroinvertebrates, which she'll tell you more about. So without further ado, I give you Dr. Cari LeRoy.
Thanks, Jim. I'm hoping everyone can hear me. If not, maybe put something in the chat and we'll try to fix it. So that was a great introduction. I'm really excited to be here. Of course, I'd be really, really excited to be drinking beers and hanging out with you all at the Swiss, but I think this is a really nice substitute. So I'm going to acknowledge my collaborators at the end, but I'm just here. I'm at Evergreen. I've been collaborating with people at the Forest Service and Missouri State, and we have funding from the National Science Foundation for this research. So it's really fun to talk to you tonight. Let's see if my advancer, of course, it's not working. Let's see here. See if I can get, there we go. Okay, so here's a picture of Mount St. Helens before she erupted. We called her Mount Fuji of the Pacific Northwest. She was a beautiful, beautiful mountain. Of course, most of us have seen her more recently in her more devastated form. On May 18th, this was just a little over 40 years ago, at 8:32 a.m., she erupted in this cataclysmic lateral blast with a huge landslide that was part of that. Most volcanic eruptions that we're familiar with are plumes of ash that go straight up. But because of the way that Mount St. Helens erupted with this lateral, this sideways blast, it kind of set the stage for some really interesting research and is really one of the most unique eruptions in kind of human history. So here's just some pictures of the landscape. These are full-grown trees that have been incinerated and laid flat by the blasts of hot gas and rock that moved across the mountain. This is part of the blowdown zone. Here's some pictures that just show, I mean, look at that. It looks like dog hair, right? These are giant trees that have been knocked down by the blast. So what happened is that you can see the heart of the mountain kind of belched forward, and it landed down below on the north face in what's called the Pumice Plain. And it pushed itself up through what's called Spirit Lake, and I'll show you a picture. So here you can see just the scale of the eruption and the blowdown zone and the blast zone and all of these different disturbance zones. Spirit Lake is there in the middle with the kind of the swirls in it. And what happened was, as the volcano slid down, it sloshed into Spirit Lake. It ran up the other side of the basin, and it pulled the entire forest down into the lake. And so here's a picture of Spirit Lake just a day after the eruption. You can see the gray surface all around the kind of moonscape, and then you see this gray surface of the water. Those are downed logs. Those are huge logs that are laid flat and floating over the surface of the lake. The entire lake was raised up by 64 meters, which is about 150 feet. And it was anoxic, meaning there was no oxygen, methanotrophic, it smelled like methane. And here's the first quiz question. How hot do you think the water got? So this is a mountain lake, right? Jim can tell us what its normal kind of temperatures are.
Cari, I think you're freezing up on us. Everybody else getting that? Maybe we can try to unshare, like not share our videos, and that might give a little bit more issues if you guys don't mind. I hate to not see your faces. We are still missing Cari. Oop. Hold tight, folks. Looks like Cari needs to come back on and share her stuff again. So I think while she's gone, the question is, how hot did Spirit Lake get? So chat your answers. I see Chris has an answer in there. Gabrielle has, oh, somebody thinks it was frozen. Other folks, I see a 145. Remember, this is in Fahrenheit. She's back.
Again. Okay. Was I here?
You were for a while, and then you disappeared.
Okay. What was the last thing I said?
So you were just asking the question about the temperature of the lake.
Yeah. Okay.
And so it looks like, from what I see, so we have our answers in. So what's the answer, Cari?
The answer is 85 degrees.
All right, so it looks like the closest person was Meg Henderson.
All right, Meg.
Yay. So make sure, sorry to butt in, but at the end of this thing or sometime during, if you can, I will give you my email address and you can send me your address to get you a pair of socks.
Cool, excellent. Okay, there's another chance to win socks in here. So, oh my goodness, let's see, technical difficulty. There we go. So here's a picture of Mount St. Helens today. You can see just how much of that, the heart of the mountain, has been moved northward. So this is a picture of the Pumice Plain right after 1980. So that kind of the north face of the mountain that I was talking about, where a lot of the guts of the mountain ended up. It just looked like a moonscape. And scientists would spend hours just kind of flying helicopters, pondering the devastation, right? And looking for signs of life, plants or little gophers or things. And so, you know, and people really thought that it was going to take hundreds of years for life to return to the Pumice Plain. So we'll see whether we think that after you see this talk. So the National Volcanic Monument was established in 1982, and it designated a certain number of acres for research, recreation, and education. So if you want to take a guess, I don't think this is a sock-worthy question, but if you want to take a guess at how many acres have been designated for research, recreation, and education, you can type your guess into the chat. But the beauty of the volcanic monument, at least to date, is that it has been allowed to recover naturally. So it's like a huge, what we call passive restoration experiment. So it's being restored just by natural forces, and we get to watch that process. Okay, we got a guess of 400 acres. Good guess. Anybody else want to hazard a guess? I'll give you a second.
I'm seeing
1980 acres. Yeah, that's a good guess. Anybody else?
I see a 20,000 from Chris.
Oh, I don't see that one. Cool. Well, it was actually 110,000 acres. So we're lucky that we get to work in this landscape as scientists and watch this recovery. Well, not really recovery, because it's coming back as something new, right? It's not the same as it was. But. So my focus as a stream ecologist is basically what you can see on this Pumice Plain. You can I can't really interact with my screen here, but you can see there's some streams that come down through the pyroclastic flow zone and flow into Spirit Lake. And so what's amazing is because of the lateral blast nature of this, it buried the forests and streams that were on the north side of the mountain under sometimes up to 300 feet of rock. And then snowmelt and springs kind of bubbled up through that. And so 5 new watersheds have formed on this landscape. They're all very small, but they're very interesting. And so Goose Creek is ephemeral, but I'll talk about Camp Creek, Geothermal Creek, Clear Creek, and Willow Creek in my talk. So I've been studying kind of how do streams evolve when they start from scratch. And it's actually a very unique location. There are not many places on Earth we can study this. So here's some pictures. This is Willow Creek and a couple evergreen students. And they're measuring sinuosity and depth and width and just kind of the hydrological variables that influence stream function. Here they are measuring stream discharge velocity. We're looking at riparian plants growing along the edge of the stream. And this is Shannon Clausen. She's a Forest Service scientist, and she's collecting benthic macroinvertebrates. So these are little aquatic insects and snails and invertebrates that live in the cobble and the gravel and the sand in the stream bottom. So this is a picture of the Pumice Plain today. So it no longer looks like that moonscape. And you can see these really neat ribbons of green, which are riparian plants or streamside plants that are growing along streams across the Pumice Plain. And so things have colonized much faster than anyone thought. So the other thing that's really interesting is that you know, we might hypothesize that it's the same moment in time, on the same mountain, receiving the same weather, the same water, flowing in the same direction, the same slope. We might imagine that all 5 of these watersheds would develop in the same way, and that would be kind of our hypothesis going into this. But the diversity of the streams that we see is really astounding. And all of these streams are only 40 years old, or some of them, you know, potentially less than that. So we see some gravelly, really kind of denuded streams that jump channels like Willow Creek that you see on the left. And then we see these marshy, mossy, kind of seepy streams like Clear Creek on the right. And then we see streams like this, where you can't even really get in there. It's just a thicket of alder and willow, and the stream is somewhere inside this nasty mess of riparian plants. So we were really interested in what drives the colonization of riparian plants, algae, which are sometimes called periphyton, and the benthic insects or invertebrates as they colonize these streams. Okay, so here, coming up, the last of these questions will be a sock winning question, but this is what's called a primary succession landscape. And so, you know, everything that's out there had to come in from the outside, maybe on the hoof of an elk or the foot of a goose or in the clouds or blown in by the wind, right? And so you'd imagine 40 years is not a very long time. So how many riparian plants do you think have colonized the Pumice Plain? So go ahead and type into the chat.
And I asked people to just text me so that, or chat with me so that, so we have 280, 50, 45, 35.
Good guesses. Yeah, so when I mean the number of plant species, it's like how many different types of plants, not how many total plants out there. But yeah, so we have 38 riparian.
Oh, so Emma was within 3. Nice job.
Okay. And then how about algae? So we have soft-bodied algae like the kind of green filamentous and blobby guys, and we have diatoms, which are these amazing organisms that build little glass boxes for their bodies. What are your guesses? So you'll have to use a comma. So guess the soft-bodied algae, comma, diatom species. Maybe I'm asking you to do too much work. There's only like one more question after this.
Okay.
Last question would be software.
27 and 18.
Oh, good.
We got 2,000. Maybe that's together. 3,060, or maybe it's 30,600. Oh, no, sorry, it's comma. 30 and 60, 15 and 20, 12 and 20.
Nice guesses. All right, it is 55 and 96 species. Sorry, Jim, I'm making you do some
That's right. I think Chris got the closest from what I can tell.
Okay. And then the last question. So this is your sock-worthy question. How many benthic macroinvertebrate species? So the number of different types of these critters out on the Pumice Plain in all of the streams. Oh, it's for a pair of socks. They're purple.
We got 40, 124, 55, 89. We got any more guesses out there?
Okay, I'm gonna let you know. It's 85. So whoever guessed 89. Probably.
Emma, once again, she also got the riparian plant species. So I think Emma wins.
Wins the socks. Nice job, Emma. Okay, cool. So there's way more diversity of organisms out in the Pumice Plain than we predicted. In fact, people told me, don't bother. You know, don't go look in those ditches. They're just like gravel ditches. There's nothing out there. You know, And then we started looking, and we were really astonished by how many different kinds of species had made their way to the Pumice Plain. So what's really cool, and this is a little bit sciencey and complicated, but if we look at 21 sites across the Pumice Plain and we look at riparian plants in panel A, soft-bodied algae in panel B, diatoms in panel C, and benthic invertebrates in panel D, and we use this method, it's called ordination, to try to place those sites in space in the graph. When they're close together, the communities are more similar to one another. We find this repeating pattern that's really kind of curious. And that is that there's this kind of group or cluster of communities, really, for all four of those groups of organisms, where samples that are at in the Willow Creek drainage or in Red Rock and Forsyth, which are tributaries of the Willow Creek drainage, are very different from the communities of organisms we see in the other three streams. And this just repeated over and over and over again. It didn't matter whether we were looking at riparian plants or diatoms or insects, we see the same pattern, which we thought was really interesting. So then we're starting to try to understand, well, why do we see those differences? Well, Willow is kind of different in a lot of ways. It's the biggest stream, it's the coldest, it has the most flow, and it has the least riparian plant cover of any of the streams. And so we started kind of trying to figure out what might be going on there. And so we narrowed in on the riparian plant cover, thinking like maybe this is a master variable. We see these ribbons of green across the Pumice Plain, and maybe we see really different communities when we have that riparian plant cover versus when we don't. The other thing is that we have a couple key riparian plants. So Sitka willow is a dominant. The other would be Sitka alder. And the willow is really neat. And so we wrote a grant to the National Science Foundation to really explore this willow and how it colonizes. Because it could be that the plant community is really helping to drive stream evolution out on the Pumice Plain. And the system is simple enough that we might actually be able to really explore that. So, so the grant was about plant sex. And you might think, well, that's kind of weird, but it turns out that quite a few riparian plants are what's called dioecious. It means that they have both male and female individuals. Most plants don't. Most plants are what we call perfect, and they're perfectly hermaphroditic, meaning that they have both male and female parts in the same flower, but not willows. Willows have male plants and they have female plants. And so we were really interested in whether this might influence how these streams work. So here's a picture of the male on the left and the female on the right. And you can only tell whether they're male or female in May when they're flowering. And so we have to get out there early. But once we started marking, we started tagging trees with pink and blue flagging all over the Pumice Plain to try to understand what kinds of patterns do we see in willow plant cover, and then how does it influence the streams. So the other major player here when it comes to willows is this little guy. Kind of cute. It's a willow borer. It's a weevil, and it bores into the stem of the plant. So it kind of like digs into the stem. Well, its larvae dig into the stem, and then the stems die and they fall over, or at the very least, all the leaves on the stem get all dried up and crispy and fall off. So this weevil attacks the willows. It was native to Europe, but it was introduced to Mount St. Helens in 1989, and it has just gotten everywhere. So it's a real ecosystem engineer out on the Pumice Plain, and it causes branch death and mortality. So we were curious about a few things, like one, does it key into the difference between males and females? And then two, how does that play out in terms of the whole system? So here you can see some willows flagged as pink or blue, and then you can see on the right a willow that's been really hammered by the weevil, in this case a male, and most of the branches are dead and all the leaves are brown and falling off. Well, I think Jim mentioned in my introduction that I'm kind of passionate about leaf litter decomposition, which is a pretty weird thing to be passionate about. But it really drives ecosystem function. It's a really key part of the carbon cycle in terms of understanding how climate change impacts might influence our world. We have to understand carbon and how it cycles. And for streams, receiving leaf litter inputs from the riparian forest is one of the most important sources of resources for everything that lives in the stream. And so when I see a system where the leaves are drying and falling in the middle of the summer, that makes me start to question, well, what is this potential impact on the ecosystem? So we did all these surveys and we found some really interesting patterns. First thing is that females tend to colonize closer to the stream. So if you're walking down a stream, it's most likely that you're in the company of a bunch of female willows and the males are further out away. And this is actually a common finding with riparian plants, especially wind pollinated riparian plants. So the males would prefer to be up in the upland kind of pollinating from afar. And the females want to be right down next to the stream because they need more resources to produce their flowers and fruits. Okay, so this wasn't necessarily shocking, but it was nice to see that there is this distinction. And then we found that females were more impacted by the weevil, which I think is also really interesting. So now you've got females growing closer to the stream. You have females getting more impacted by the weevil, dropping their branches and leaves into the stream. Now you can see why I'm getting really excited, right? Okay, so we take all these willow leaves from across the Pumice Plain and we put them into little mesh bags, and then we incubate them in the stream to look at leaf litter decomposition dynamics. And what we found is that the male willows had higher nitrogen, so their leaves are tastier. And so the female leaves were less tasty, and so they decomposed slower than the male leaves. And this is important because then the female leaves are a longer term resource in the stream. If they last longer, basically, they're a resource for longer. So, and they're colonized by different benthic macroinvertebrates, which is really interesting. So the benthic macroinvertebrates might be keying into things like nitrogen or defensive compounds like tannins and other things like that. So, so that's one way that female willows and male willows differ. The second is I had a student out there named Iris Garthwaite who noticed all of these willow flowers in the stream in the summertime. And she started wondering, well, what kind of a resource are these willow flowers for these organisms? Do aquatic insects eat them? Do they live on them? How nutritious are they? And so she did a whole study that was funded by the Evergreen State College on these willow flowers, and she's getting ready to publish it. And so what she found was pretty interesting. So she put flowers in one mesh bag and she put leaves in others and put a bunch of mesh bags out. And what she found was that the flowers show this kind of like steady rate of decomposition through time. And the leaves drop, they lose a ton of mass early on, and then they decompose very slowly. And so they're really different resources for streams. She also found, and it's hard to see, but this little picture in the bottom corner is it's called a caddisfly. And a caddisfly is a little aquatic insect that builds a case for itself out of whatever it can find. And she found several of these little caddisflies that were actually building their cases out of willow flowers. And so not only are they potentially a resource in terms of eating, but they're also a construction resource, like a source of materials for caddisflies. She also found that, and this is another one of those weird ordination graphs, that the communities of aquatic insects that colonize flowers are significantly different than the communities of aquatic insects that might colonize leaves. And so again, more evidence that these willow flowers are influencing stream ecosystems. And again, the females are making these particular types of flowers. So to kind of conclude this part of the talk, males have higher nitrogen, lower carbon to nitrogen ratio, so they're tastier, they decompose fast, but there's fewer male leaves in the stream because they're colonizing further away and they're less infected by these weevils. So they're not dropping as many of their leaves and branches into the stream. They also produce smaller flowers, and so they're not producing as much of a resource. In contrast, the females have low nitrogen that they decompose slow, so they're a longer term resource. They colonize closer to the stream and they're more infected by weevils. And so there's more female leaf litter in the stream, more likely to drop their litter in the summertime. And that litter that they drop in the summer, I don't think I'm showing the data necessarily in this talk, but it differs from in terms of chemistry from the litter that falls in the autumn and from male leaves. And that female flowers host more insects than leaves. So a couple other things that are just kind of fun. Basically, Mount St. Helens is inaccessible from about October through May. And so nobody knows what's going on out in those streams unless you like ski out there, and almost nobody does that. So we installed these wildlife cameras so that we could spy on them. So here is kind of a life from September to I think about May for a stream. You can see, I don't know if you noticed that big pulse of sediment. And then we're going to get, snow melting, snow, just kind of like this snow coming in and out in the stream. And then you're going to start to see the willows greening up. See, isn't that cool? And the alders. And if you were looking really closely there, I think it was an elk. And oh, there were my students. So I think I can let you watch that again. I think it's just going on its own. I'll have to stop it. So here we go. You can, oh, no, I'll start it again. Sometimes it's fun to watch it. So look for the elk in the upper right-hand corner and look for the cameos of the evergreen undergraduates and watch for the sediment pulse. Whoa, did you see that? So, I mean, this is like nobody really knows what's happening to these streams, how much flooding there is, how much snow there is. We just show up in May and we're like, whoa, now the channel's over there, right? And so it's been really fun to have these cameras. Okay, look for oh, did you see the elk? And then there's gonna be some people, there's people. Okay, all right, I think that's almost done. So the last thing is we've been learning to fly drone. So it's a no drone zone unless you have a permit. So through my research permit, I've been able to fly. I don't know if that's loud enough. Can you hear it? Oops. Oh no.
I don't think we got to hear your audio, but the visuals were really cool.
Well, I can make a, I can post the YouTube link in the chat after I finish. I've just got 2 more slides and then
Cool.
I'll do that. So, oops, let's see, go back. So I've really just had such a great time at Evergreen working with undergraduates on this project. Lots and lots of undergraduates. We call them Wondergrads. They're terrific field assistants, laboratory assistants. They're the people that are sorting through and identifying aquatic insects and going to conferences and presenting posters. So we've had a lot of fun. Funny thing is, I think everyone you see here, maybe a female. I had the uncanny luck of just having this massive female field crew the last 2 summers. And so I just want to thank all of these amazing students. And with that, I'll thank the National Science Foundation and the U.S. Forest Service for funding to support my collaboration with Shannon Clausen and Charlie Crisafulli for logistical support, as well as our permit to work at the mountain. The Evergreen State College has provided a lot of support, especially through its summer undergraduate research fellowship program. Dr. Deb Finn is at Missouri State University, and she brought students out last summer. Dr. Dylan Fisher at Evergreen for technical support, a bunch of people for field assistance, and Mount St. Helens Institute for allowing us to participate with the summer ecology program, Upward Bound, and the Geo Girls program. And then I'll just end with a little note that gets a little bit into advocacy. There is a motion on the table with the Forest Service to construct a road across the heart of Mount St. Helens, right across the Pumice Plain. It would cross every single one of my streams, and it would really kind of put a stop to the research, but it would more important than that, it would really change the development, the way that these streams are developing. So if you feel strongly that there should be no road, there are ways I think that you could contact the Forest Service and encourage them not to build their road across the mountain. So with that, I'm excited to take your questions, and I will, I'll close this out unless people want me to go. I can go back to slides, but I can close this out and then I can get the link to that drone video for people to watch. So thanks very much for your time.
Thank you, Cari. It's a visual round of applause for Cari. We have several questions for you. People are very interested. So you ready for the first question?
Sure.
All right. First question comes from Chris. He asks, do willows and alders have mushroom fungal symbionts to help them absorb nutrients? Any studies on them in this zone, if so?
Yeah, so willows are definitely mycorrhizal. I can't remember if they're ecto or endomycorrhizal. Alders have a complex relationship with a nitrogen-fixing bacterium in their roots. And so both are provided with extra kind of resources through these symbionts. Alders can colonize almost any habitat. It's very likely because willows are in Salicaceae, which is the same family as poplar. Actually, there's a professor at UW in Seattle named Sharon Doty who has found nitrogen-fixing bacteria, not in the roots like we're used to with peas and beans and things like alder, but in the stem of cottonwood trees. And so it's very possible, because she's been looking in all kinds of plants, that the willows out there also have a relationship with nitrogen-fixing bacteria in their stems. So yes, definitely mycorrhizal and possibly nitrogen-fixing.
Awesome. Next question is from Emma. They ask, what kind of data were you collecting with the drone? Any LiDAR by chance? Ooh, somebody's in the know. I teach in our geospatial curriculum, and many of our students are interested in LiDAR collection via drone.
Yeah, unfortunately, my drone is just an RGB drone, so it's just light, just pictures. But I am kind of creating these big ortho mosaics, and I'm trying to look at change over time. So I'm especially interested in like channel changes just between, the spring of one year and the spring of the next. It's a lot more difficult to fly out there than I expected. It's great because there's nothing in your way, right? There's no obstructions, but the weather it never cooperates. It's always so windy out there, or it's like socked in and foggy. And so the drone has been a challenge, but I'm excited to learn, and I'd be happy to share, images with students if people want to do anything. Like, I think there's a ton of opportunities for just making measurements of things, at that scale. So yeah, I'd be happy to talk with you more about the drone imagery and like what we might be able to get out of it, because I'm a total newbie when it comes to drone flying. Although I got my license, so I'm official, but I just don't.
Nice. Do you get the little wings with that?
I wish. I
That
should
would be
order
nice.
myself some wings.
Uh-huh. Actually, kind of follow up on that question, can you, with your stitch together thing, can you actually see 3D, like estimate? I'm thinking of like all the amount of sediment that comes down, if you can actually estimate changes in sediment, because we've seen years where you come back the next year and the stuff that I put there is actually completely buried by sediment and I can't find it anymore.
Yeah, I think it does give me a digital elevation map. And so I think there's some limited 3D capability with the ortho mosaics. Yeah.
Cool.
So yeah, we should look, especially if they build the road. That's the other thing is I'm excited that I got out there to do a lot of imagery pre-road, because I think if they build that road, it's going to really change things. So they're not going to like the fact that I have pre-data, but.
Uh-huh. It's always good to have pre-data. So another question from Chris, are salmonids spawning in these creeks, and do they also dump nutrients?
Great question. So unfortunately, and Jim knows more about this than I do, there were, we think, some rainbow trout that were introduced by maybe just a random citizen into Spirit Lake. And those rainbow trout do come up some of the streams, but not all of them. And so again, there's kind of this gradient, so that you'd think that they would come up all the streams, but because there's so much variation in water temperature and flow, some of the streams just are better habitat for the rainbow trout. And yes, anytime you have fish, you have all these kind of fishy inputs that influence nutrients. So for sure.
Cool. I had a complex question. So because of this relationship between the stream banks and the different sexes of trees, what happens when the stream moves? Do the females that were there next to the stream, do they start dumping more material? Does it switch over? Like, I'm interested in, like, are you suddenly like populating the open area with females because the stream moves?
That is so funny, because on Saturday, I had my students look at that, because we were out there with our masks, being socially distant, and I said, you know what? Willow Creek had made a huge jump again. It does every year, right? And I said, well, what about, let's go survey this. There was kind of a bar of willows that I knew had been separated from the stream for quite a while, at least the last 4 years. And I thought, what if there's differential mortality? What if, okay, we know maybe it was female dominated when the water was right there, but after the water leaves, do all the females die, right? And so we went back, and unfortunately, a lot of the willows were either so young we couldn't tell whether they were male or female, or we were too late in the season to tell. They already dropped their male flowers, but we surveyed about 100 willows, something like 80 of them we couldn't tell, but we only found one male, and we found like 20 females. So possibly the females can hang on even after the water leaves. But that's a great question, and I think we should do more research to follow up on that particular question.
Excellent. Well, I think that was the last question. I don't see anything else unless somebody chatted to you directly. I think they were all pretty good about sending folks questions my way.
Okay, I don't see.
Okay.
Oh, it says there's only an option to message me or you separately, not everybody.
Yeah, so they were all, actually, I did just get one last question in. Oh, sorry, two last questions.
Well, and I might have gotten some responses to my questions. I got anyway, the socks. I don't think, only a couple people wrote questions, like those kinds of responses to me. So.
Gotcha. So Grace asks, does this correlation between the willow sexes and the streams work with the hummocks ponds too?
That I don't know. And those hummocks ponds are so cool. Yeah, Charlie Christopher's been studying those ponds for quite a while. I don't know if, Jim, you've gotten roped into that, but he's roped all kinds of people in. And I don't think anybody's looked at whether there's male or females or how many of each. I don't think there have been any surveys done there. But I don't know. Maybe they've done it in the past couple of years. Not that I've heard of.
Yeah, not that I know of. And it'd be interesting because for those of you who don't know, the hummocks ponds are this whole set of, God, I think there's 100 or so of these things that got created during the whole landslide and debris avalanche. And so they're all new as of 1980, but they dry up. So there's actually a bunch of them that stay wet all year and other ones that dry up by the spring, some that dry up by the summer. So that idea of instead of the stream moving but the water disappearing would be interesting to see if you've got your similar pattern in that sort of situation.
That is a cool question. The other thing that's nice is those hummocks ponds are actually pretty accessible in May when you need to be there to see the flowers. And the Pumice Plain is really a pain to get to in May. You have to hike like 14-mile day to get out and back and get all your work done. So it's no small feat. But yeah, maybe the ponds. Whoever has that question, you should email me. We can go survey next May. That sounds like fun.
And then last question for you: how did they learn that the weevil was introduced to Mount St. Helens in 1989, and were there any repercussions for this person's actions? Do they know who did it?
I don't think we know who did it. And I think the only way we know about when it showed up is when people started seeing it. So Charlie or John Bishop at WSU, Vancouver probably first noticed it. It's similar with the trout. I don't think we know who did it. We just know that at some point we started seeing trout. Kind of like, unfortunately, and I didn't mention this in my talk, but in 2016, because of trout that Charlie and probably Jim had caught, in their stomachs, we started finding New Zealand mud snails, which is a class 3 invasive species in Washington State. It's like nothing you want to mess with. And unfortunately, the snails are starting to make their way up some of our streams. And so we have to be really careful now not to go to certain parts of the stream and walk around because we might spread the snail. So again, like if you're out there and you're sampling or measuring things, and in 2016 we started seeing, we started sampling in 2015, and it wasn't until 2016 that we started seeing snails in our benthic invertebrate samples. So yeah, you just have to kind of make observations until you notice something, unfortunately.
Well, let's have another silent round of applause for Cari LeRoy for an awesome talk. Thank you very much. And hopefully, thank you all for tuning in tonight. Make sure you let folks who couldn't make it know that this is going to be available at the Grit City Think and Drink site. Hi, Grace. Some people that I know, so definitely we hope to see you at our next one in August, talking all about gerrymandering. And I would be remiss as a scientist if I didn't tell you to make sure that you mask up when you're going out right now and show your Tacoma pride. So if you want to get some awesome Tacoma face masks, you can go to the great people at T-Town, and they are selling these. They're really awesome. So show your pride, wear your mask, be safe, and it's been awesome to see everybody. Everybody have a great day, and thank you very much, Cari.
Yeah, and if you hang out, I'll get that YouTube link if you really want to see the video again. But thanks for everybody. It was really fun to meet you all.